In a clinical setting, patient presents with some sort of microbial infection. Hey, great, just throw a broad spectrum antibiotic at them. That'll take care of anything and everything. And that's used to be how they thought about it when they first came up with, antibiotics, what we think of nowadays as antibiotics, antibacterials. Just take the biggest hammer and, that's what you hit it with. Well, nowadays we need a little more finesse. So what we want are antimicrobials, antibacterials that, are more of a scalpel. Instead of a sledgehammer, it's more of a scalpel. All goes back to, where do we get these antimicrobial agents? I touched on it in the last lecture. Bacteria and fungi have been fighting each other for a billion years or more. They've been fighting each other, evolving methods for attacking each other. They can't physically attack each other. That's just not, it's single cell organisms. What they can do is they can make compounds, chemicals that they excrete into their environment that will have no effect on that cell itself. But if any of its competitors come into contact with it, that chemical, that compound, it will damage them. It will damage or it will kill them. These are the natural antimicrobial compounds that we've found. I used penicillin as a great example of it. These are bacterial or fungal derived. Different bacteria. We find soil microbes are great, have been a great source for many of the antibiotics that we now currently use or have used. Fungi, great source. In a clinical standpoint, you have a patient that has an infection. You need to start treating it. How do you do that? Well, as I said in the last lecture, you could throw the sledgehammer at them. Drop the bomb. Here's a broad spectrum, everything's taken care of. Well, that could result in greater issues later when you've wiped out all the competition. All the microbes that should be on our skin, should be in our gastrointestinal tract. All the good bacteria, gone. Anything I get exposed to now, whether on my skin or in my gastrointestinal tract, better hope it's not something that could hurt me, because there's no competition now. They're going to overtake. So in a clinical setting, things you want to do is you want to identify so that you can start targeted treatment. Okay? Targeted treatment. That is now the key to medicine, to clinical treatments in this day and age. So first, you have to ID. You have to figure out what the microbe is. You'll take a swab. You'll take a sample of some sort. Aspirate off some of the pus, some of the liquid. You'll send it off to the ID lab. The ID lab will do many of the tests and stains and things that you're doing in the virtual labs right now, or you're set up for you to do over the next few weeks, based on gram stain, based on physical appearance of the cell, based on biochemical reactions, the different bacteria that could be or would be present there. Think about it. You scrape, you aspirate, whatever, you swab. There's going to be many, many different types of microbes there. The majority of them should be there. The ID lab's looking for the ones that shouldn't be there, because the ones that shouldn't be there, pretty good chance those are the ones causing the infection. So they isolate, they identify based on, like I said, gram stain, cell, cell shape, whatever, in biochemical assays. Here's the microbe causing the disease. What they'll then also do is number 2, susceptibility. What antibiotics work against this microbe? What antibiotics at what concentrations. Giving a quick identification that could be done within 12 hours. Figuring out susceptibility, that's going to take another 12 hours or so. So that's why in a clinical setting, if you've ever been in the hospital because of an infection or family member or you're a clinician, you'll see that over a couple of days time period, they can change what antibiotics are being used. They can change what concentration as more information becomes available from the lab. And then condition of the patient. Can the patient handle this antibiotic? That's why they always ask, do you have any allergies? Are you allergic to penicillin? Are you allergic? Are you allergic? Are you allergic? Is the patient on the verge of death? Are they about to succumb to the infection? If they're about to succumb to the infection, don't worry about trying to find a narrow spectrum antibiotic. Use the sledgehammer. Use the broad spectrum antibiotic until, keep them alive. Last lecture, I used the toxicity one. Yes, we have a silver bullet that can knock this out, but it has a high toxicity level. Hey, patient's going to die in 24 to 48 hours from the infection. Nothing else is working. Let's use this one. Condition of the patient. Now, once you've identified the microbe, second thing you need to do is test susceptibility, because you do not, as a clinician, you do not want to prescribe an antibiotic that is going to have no function. It has no metabolic activity against the microbe. So you need to know which antibiotics are going to work and what concentrations needed. Because again, things, the antibiotic might work, but if you're prescribing at too low a concentration, it's only going to wipe out some of the microbes, and those that it doesn't affect, are going to be the new population causing the infection, and within a few days, the antibiotic is going to have no effect whatsoever. So you want to correct antibiotic, correct concentration. Those are the keys. So you need to know, for the microbe that the ID lab has determined is causing the infection, what is the level of resistance against the known antibiotics that would generally be used. Staph aureus in me, because of my history, my medical history, what I have eaten, what I have drank over 49 years, means my staph or whatever is in me, on me, it's going to be different than what's on you, because you haven't drank the same stuff that I have. You haven't come into contact with the same people I have. You haven't had the antibiotics I have over so many years. So just because it's the same species of bacteria doesn't mean that what you have and what I have are going to have the same level of susceptibility or resistance. So that's why they have to test this. You look here, the picture of the Petri dish. It kind of looks all clouded over. That opaqueness is actually bacteria growing on the surface. They've spread it out. Not doing that isolation streak that we've talked about in a previous chapter. They've spread the bacteria all over this. They call it a lawn of bacteria. There are so many colonies there that when they're clumped together, they look like a lawn of grass. You look at the grass, you get up close, you see its individual strands, blades of grass, but you get back far enough away so you get the big picture look. It looks all even, completely covered. Same here with this lawn of bacteria. The little white discs you see are paper discs that have been soaked in different antibiotics at different or same antibiotic, different concentrations. That zone you see around, that's what they call the zone of inhibition. The Kirby-Bauer test is a test to see, does the antibiotic work against the microbe, and is that microbe susceptible or resistant? That zone of inhibition, basically, It's an area around that disc where the antibiotic has leached out and then diffused out of that disc, diffused through the agar. Further away from the disc you get, the lower the concentration of that antibiotic. And what you see is the bacteria grow up close to the disc as they can, because they can overcome the effects of the antibiotic. They're resistant to it. The closer they get, the more susceptible to that antibiotic at that concentration. So you make the lawn, you put the discs on, you incubate it, and then you wait. As the bacteria grow, increase in numbers, they'll grow up close or far away from the disc. Go through with a ruler and you measure the diameter of that zone of inhibition. They have tables where they have gone through, and for every antibiotic, bacitracin, chloramphenicol, erythromycin, gentamicin, neomycin, penicillin, whatever, and for each microbe, this one shows the Kirby-Bauer test for Staphylococcus aureus, how close or what diameter that zone of inhibition should be. And you see they're susceptible if the diameter is greater than this. They're resistant if the diameter is less than this. It means if they're resistant, they can grow closer and closer to that disc because they can handle higher and higher concentrations. Problem is that takes time, and doesn't always, it's dependent upon what's the initial concentration of the antibiotic in the disc, what the zone's going to be, and it's not very definitive. Susceptible, resistant to this disc doesn't tell you what concentration is that boundary of susceptible and resistant. It doesn't give you the minimal concentration necessary. So what they've developed a few decades ago is what's referred to as the E-strip you see here. The E-strip is again a strip of paper, and you can see, if you look closely at the figure in the textbook, you'll see it has markings on it, delineations, to show, as it goes up, the concentration of this antibiotic is higher and higher the further up you go. How they make it, don't worry, they just do. So down at the tip, you see there's very little. Up at the top where the E is the highest concentrations possible. So you see, instead of a nice round zone like you get with the Kirby and the little discs, you get this spear-shaped, arrowhead-shaped, high concentration, greater zone of inhibition, lower concentration, grows right up to the paper. So what you can do is you can go through and you can get an estimation within this area where you start to see it getting wider and wider and wider. It'll tell you, this concentration is susceptible. Further concentrations below this, it grows more and more resistant. Still old school. Still doesn't help, give you definitive. So what we have nowadays is we utilize test tubes or 96 well plates. The test tubes, we can tell growth, no growth by whether or not it gets cloudy, and we can put in a specific concentration of antibiotic so that we know that so many milligrams per ml of this antibiotic will inhibit or not bacterial growth. Turns out test tube's big, bulky, takes up a lot of space. Unless if you're going to do lots of dilutions, that's a lot of tubes. Every dilution's another tube, and trying to do stuff and duplicate, triplicate, so it gets to be a bit unwieldy. So what we do now is we use the 96 well plate. You can go through and you can do a serial dilution. high concentration to the far left, and then every well as you go to the right can either be half the concentration, a tenth of the concentration, whatever you want it to be. And you just do this serial dilution. Take a little out of the first, put it into the second, mix it, take out, same amount, put it in the third, take out, put it in the fourth, put it in the fifth, the sixth. So that way you could know that, if it's serial 2 dilution, every well as you move from left to right is half the concentration. Boom, 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 boom. And then you can put in some dyes. And you aliquot the bacteria in, and then you just let it incubate for a few hours. Since it's such a small volume, it's not going to take that many rounds of bacterial division till you start to see some effect of the metabolic waste turning the dye a different color. So then you can just sit there and see. Where's the growth? How much? Where's it at? You see where it's blue or purple, Between those 2 wells tells you specifically, this is the concentration that you want to be at. Because before this, it can grow. After this, it can't grow. And that's just how it works. So you can go through and you can calculate based on the number of wells, whatever dilution you're using. Here's what I started with. It's diluted 6 wells at a half dilution each time. So that's 2 to the 6th. 2 to the 6, 2, 4, 8, 16, 32, 64. So it's 1/64th of what I started with. And there you go. The 96 well plate, since it's small, compact, and only a few hours of growth compared to the test tubes, which if you double that time. The Petri dishes with the E-strips or the Kirby Bauer, minimum 24 hours. The 96 well plate's the way we go now.